one chapter, one topic a day. So that should help you to improve your skills. So today, what we have to do is, I hope you can see the PPT presentation. If you have any problems there, please put it in the chat box. Okay. Excuse me. Sorry. Yeah. So we were talking about the structure of DNA, and we also see who proposed the DNA model and who performed the X-ray crystallography, right? So we talked about it. So to summarize the structure of DNA, so we know that it is a double-stranded polynucleotide molecule. So I told you that sugar and phosphate. Okay. Hold on. Excuse me. Perfect. I'm getting a pop-up, so which is not usual. 1 second. Yeah. Anyways, so hold on. Yeah. We're talking about how sugar and phosphate makes the backbone of the DNA, correct, right? And the bases project towards inside. So they are right-handed helix. So they go in an anti-parallel direct. Sorry, they are right-handed helix, and you can see that it is in a staircase module. So the strands are having a polarity. That means one strand goes from 5 prime to 3 prime direction. Other strand runs from 3 prime to 5 prime direction. And the diameter of the DNA is 20 degree angstroms, and then you have the base complementation. I told you what is complementation in the last class. That is adenine always goes with thymine, and then you have cytosine that always goes with guanine. So it is never ever the case where the nucleotides mismatch. It's not possible. Okay. So there is a very serious base complementation, I can tell. Okay. So that is happening. Now, you understand, like usually what happens is, I told you in the last class, whenever you find a sequence of codes like this that is given in a question paper, especially NEET question paper, there are two types of questions that might come. So what they will do is they will write 5 prime to 3 prime, and they will give this particular line, let's assume. And then they will ask in the question, write the proper sequence of what is the proper sequence of mRNA. So out of these, what will be the mRNA that will be produced out of this strand? Okay, so that they might ask. So that is the first thing. Or they might give both the strands, like 5 prime to 3 prime and 3 prime to 5 prime, and they might ask, out of this strand, can you tell which what is the sequence of mRNA that will be produced? So this direction, observing the direction of the strand, is very, very important. And we will see later on why this polarity, like 5 prime to 3 prime and 3 prime to 5 prime, is very, very important for us to observe. You will know the concept, but keep this observation in your head. Okay, that is first thing. Next thing, what we understood is in each turn, okay, the whole distance is around 34 degree angstroms. And with each turn, what do we have? In each turn consists of 10 base pairs. So if we just divide this 10 with 34, you know that it gives 3.4 degree angstroms. So is it that precise? Is it always, 10 base pairs and the distance between is 3.4 degree angstroms? To be very general, yes. To be very general. But if you go very specific, 10 base pairs and around 3.4 emissions, we can see that in each turn we have 10 base pairs. Okay. And the distance between the base pairs. Hi everyone. Sorry. I don't know. I think I lost an internet. I hope you guys can hear me right now. Can you hear me now? Can you confirm, guys? Can you hear me? Yeah. Sorry about it. So yeah, I was just summarizing about the structure of DNA. I don't know till where you heard, but you can just let me know. So at some point we can reconnect. Okay. So let me go to the next slide by the time you guys text me. So this is the important part. Till now, the last class we have covered. So very important part is, are there any other types of DNAs that we can find? So the answer is, if you see in the nature, we can only find majority of the organisms from bacteria all the way till complex eukaryotes. You can find only one and similar type of DNA that is called as a bDNA. So what is a bDNA? bDNA is the DNA module that your Watson and Crick proposed. Okay, are you following me? So they proposed what? The bDNA model. So now you know that sir, we can't see slides. Is it true for everyone? Okay, chalo. Hold on. I really don't like this sometimes, you know. Now I think you can see the slide, guys. Correct? Yes, yes. You can see the slides. Mm, yeah. So please tell me if you can see the ink. Hello. I just wrote hello. Can you see? Please confirm. Yeah. So I hope you can see, guys. And I'll also check if there is any problems. Okay, I'll just continue. Okay. So one important thing that we have to remember is there are three types of DNA. Okay. Okay, change to yellow. Color only, no? I'll change it. Give me a second. So, oh God, thank you for being patient, guys. You know, yeah. Hi. I think you can see now. Yeah. Yeah. So there are three types of DNA. One is aDNA, bDNA, and zDNA. So as I was saying, this bDNA is the one that we can find in all living organisms, and this is the module that even Watson and Crick worked on. That is the bDNA. Okay. Now, you know the properties of the bDNA, that it is a right-handed helical DNA. So the distance between the each base pairs, I mean, like in each turn, there are 10 base pairs, etc. Right? So what are these other alternate forms of DNA? So a stable molecule like our bDNA can also exist as aDNA. So aDNA is not natural. Okay, it is not natural. So what is the meaning of it is not natural? It cannot be found in nature. We can artificially make aDNA. Oh, what is artificially making aDNA? Now, what we do is we take the bDNA, okay? We take the bDNA, and now what I will do to this bDNA is I will dehydrate, okay? So when I dehydrate my DNA molecule, now what happens is I will lose a lot of water molecules. When I lose a water molecule in my molecular structure, now it will try to get compressed. So the aDNA is also a right-handed helical DNA. Remember this. This is a very important term. So I'll write it as right-handed. Okay. So right-handed helix. So what is a bDNA? bDNA is also a right-handed helix. aDNA is also a right-handed helix. The significant difference comes in the arrangement of the nucleotides. So in bDNA, we know how the nucleotides are very nicely arranged, like 10 base pairs per turn. But as we dehydrate this, now what will happen is the compressed form of bDNA, I can tell, because of loss of water. So when we lose water, the structure gets shrunk. So now in each turn, what you can find is you can find more than 10 base pairs, in each turn of aDNA. So what are the two things that we have to remember? That aDNA is not natural. So aDNA is a right-handed helix. So when you dehydrate the bDNA, you will get an aDNA in laboratory. You get my point? But when it comes to zDNA is a very, very unique type of a DNA. Okay, zDNA is very, very unique type of DNA. So what is the most font size, guys? The information is in the written slides. So please adjust for today's class. Okay, I'm losing time. I will try to make sure that these slides and the notes come to you. Okay, no pressure for you guys. So yeah, when it comes to zDNA, see guys, in zDNA, what happens is when you see some of the scientific proofs in some plant cells, very rarely the zDNA has occurred. Okay, now what is zDNA? Z is like zigzag, right? So if you see the zigzag, you know what is a zigzag? So it is like this, and here, 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 here. So that is called as a zigzag. So basically the structure is like alternating zigzag, zigzag. So that is one thing. When you look at it, that's how it looks like. So now this also has a turn. You can see here, right? So this is a turn, this is a turn, and this is a turn, and here is also a turn. So if I look at my, if I look at my ZDNA, what I can see is the turn is not right-handed, it is a left-handed. Okay, it is left-handed helix. So that is first thing you have to remember. So what is the DNA? It is a left-handed helical DNA, and the DNA is zigzagly arranged. And if you look at the arrangement of your purines and pyrimidids, you know that, right? Our nucleotides can be divided into two important categories. One is purines, other one is pyrimidids. So you know that the purine is adenine and guanine, correct? No, your adenine and guanine. I'll write it as and guanine, and your pyrimidines are cytosine, thymine. Okay, so these are your pyrimidines. So these purines and pyrimidines are arranged in alternate fashion. Very, very important point. In which DNA structure? Alternate. Okay, so alternate purine and pyrimidine. Got it, guys? So what is that meaning? For example, if I have adenine here, you know that this is thymine. So adenine, thymine. Done. Next, this is a purine, correct, right? Next, pyrimidine. What is pyrimidine? C, like this, or it might be T, or it might be G. So a purine, pyrimidine, like this. It's arranged in a zigzag form. So what do we get out of this slide? First thing, in most living organism, what type of DNA is found? bDNA. This is a compulsory question. I mean, like most recurring questions. That's why I'm insisting. Listen to me. Which is the most common form of DNA found in living organisms? That is bDNA. What kind of model did Watson and Crick propose? That is also bDNA. Next one. Now, which is the DNA that is synthesized that can be synthesized in the laboratory? The answer is aDNA. You understand? So the answer is aDNA. What type of DNA is aDNA? It is a right-handed helical DNA. Okay. What type of DNA is zDNA? It is a left-handed helical DNA. Similarly, so these are the pointers I have given. Based on these pointers, the questions can be made. So make sure that you remember the features of this particular DNA and the differences between them. I hope you understood what I just thought. If you have any questions, you can put in the chat box. Okay. So chalo. Now, the greatest thing about Watson and Crick is they not just propose the structure of DNA. During the proposal of the structure of DNA, they made they made three significant points, guys. What is that? So they said that always adenine goes with thymine and cytosine goes with guanine. That is, they told the base complementation. Correct, no? They always told A goes with T and then C goes with G or G goes with C. So this is what they told. This is the first one. Correct, no? And the second one, what they told is the semi-conservative nature. That is, they predicted, they did not prove. They said that the DNA is replicating in a semi-conservative model or semi-conservative model of replication. That is second thing. We'll come to this point. And the third important point they said that is DNA to RNA and RNA to protein. So this chain is possible. It was predicted or it was suggested by Watson and Crick, but it was not proved by them. So what is central dogma? Central dogma is the flow of life, I can tell. What is the meaning of flow of life? See, when an organism is born, these organisms have to grow, they have to metabolize, they have to reproduce, correct? No? And then they have to age and die. During this process, you can see that even a human being, to any organism, the life starts from one single cell. So from one single cell, see, my life started from where? One single cell. Now, how many cells are there in my body? There are around 30 trillion cells. Okay. So there are around 30 trillion cells now. Are all the cells same? Definitely no. They are not same. Do they do the same function? Definitely no. They have their own labor. They do their own work. They don't interfere much, but they have the coordination. So who tells this? What is this information? It's like you have a phone and the phone has to perform. So for that, what you need is you need something called software, some information that is stored that can behave in such a way, correct, right? So our nature, what it does is it stores this information for the expression, like whatever you become, the way you look to the way you are, everything, all the characteristics that you are is already encoded in your genetics, that is in your DNA. You understand? So this DNA, we have the stored information. How does this information gets conveyed, or how does it How does it, for example, you have a cell, guys, okay? You have a cell, and the cell has nucleus. Nucleus has DNA. Now, this genetic material which is present in the nucleus has to make these cells work in a certain fashion. So, what it has to do, it has to have a chain of command. It has to tell, see, you, this is the information. I'll transfer the information to you. You take it to the cell organelles. Ask the cell organelles to construct, okay, a certain thing that I am asking in a particular fashion. So, this is what is happening. So, the the messages that are coded in your DNA in the form of what? Genes. Correct? No? So what are genes? We are studied in principles of inheritance and variation. They are expressing. Correct? No? So that is something that gives you character. Okay? That is called as gene. So these genes which are there in your DNA, what they do is they create a message, okay, with the help of other proteins. What kind of message they do? They create in the form of RNA. Let's say that it's a very generalized function of RNA. I'm not telling that this is the only function, but major function is it carries the message from the written in the DNA. It is converted to a form, and that form is called as mRNA or messenger RNA or just RNA. Let's call it as RNA. So this translated message that is in your DNA or transcribed message that is in your DNA is taken from the nucleus and it goes to the cell cytoplasm. In the cytoplasm, what happens? Some of the cell organelles, they come and decode the messages that is present in the DNA, and finally it results in the formation of protein. Now you might think, protein, we have heard this term called as 1,000 times. We all know protein. But what is the significance of proteins? Why it is converting into protein? You see, more than 98% of your all biological functions, okay, all biological functions is done by proteins. Okay, your repair, regeneration, growth, signal, transport, all of them are protein. Yeah, protein. Okay, protein is involving in all of these things. So that is why. So this is the flow of life. That means flow of information within the cells, flow of information within the organisms. So this flow is called as the central dogma of life, or it is also called as central dogma. So what is the sequence of central dogma? You should know that it is DNA, RNA, and RNA to protein. Correct? No. The process of converting the DNA into a transcribed RNA. So this process of DNA to RNA, correct? No, is called as transcription. Whereas from RNA to protein, it is called as translation. Now, don't worry if you are not so much comfortable with transcription and translation terms. It might find very much for you. As we study in detail about what is the process in transcription and translation, you will get more aware of this term. Correct, right? So remember, guys, this direction and then terms transcription, translation, and replication is very, very, very important. Okay, is there any doubts? I hope you can see the slides and everything. Okay. Next one is the packing of DNA. So now why the DNA has to be packed? That is the most important question. And why are we even talking about? We studied the structure of DNA. Excuse me. And we also studied that there is a flow of information in life, so which happens through central dogma. Now, next part is the most important part, the transfer of this information. That's what it comes. Now, if you look at an average organism like the human beings, we can see that our each cell, okay, each cell contains around 6.6 billion pairs of our genetic material or 3.3 billion base pairs. Okay, so each cell has around 6.6 billion bases and or 3.3 billion base pairs of nucleotides in each cell. You understand this point? It's very large. Imagine around 3.3 billion monomers arranged in a sequence. Such big molecule, you know. So that is the information that you have in every single cell. Okay. Now the same goes with other organisms also. It can be even a single bacteria, or it can be an elephant or a bird or anything. They have a lot of information. More than the volume of information, that is how much huge this information is, how important is this information, correct, right? It's very, very important. Imagine a small, guys, a small nucleotide chain we saw in the mutations in the last class. One base pair change happens, the whole expression of the body changes. So that precise it should be. What our life thought, that means our cells, our ancestor cells, no, we all came from the same thing. So what they thought was this information I am having is very, very vital. Why? I have to make sure that I transfer it to the next generation. First thing, correct? No, if you have one organism, what it will do, it will reproduce at any stage of life. So why reproduction? It has to make sure that its genetics pass from one generation to another generation. So this is a very, very key aspect of biology, I will tell you. Now, during this process, when I am giving the information, I have to make sure that it is as safe, as sound as possible. That is when the packing mechanism comes. So the genetic material, the DNA itself is not alone. It is accompanied by some of the proteins which help in precise packing of the DNA material. The DNA is packed, okay, in several ways. If you see the prokaryotes and eukaryotic cells, the complexity of packing or how these molecules are arranged is completely, you can see that it is a lower version and it is a higher version when you compare between prokaryotes and eukaryotes. So this packing was done to make sure the genetic material is safely transferred during the cell division. Okay. The division of the genetic material happens properly. The transfer of genetic information happens properly. For that, genetic material has to be packed. You understand? So the reason for packing. Now, the packing happens in two ways. One is the packing in prokaryotes we see, and then we see the how complex it is in eukaryotes. Now, prokaryon, the term itself will tell that it is a primitive cell or it is a primitive nucleus. So, what is the meaning of primitive? Earliest. So, the earlier version of the phone is like the less advanced, right? Now you buy is the most advanced, like that. It's just a simple example. So, they have a early nucleus. That means the nucleus or the cell is not much evolved. It is a beginning cell. So now what happens if you look at the cellular characteristics of prokaryotes, you can find that they don't have definite nucleus. Meaning is the nucleus is not organized with a membrane. They don't have any wall or a bubble for a nucleus separately. So nucleus is just naked. If you see the cell, you know this meaning because you have studied the cell structure. Correct, right? The nucleus is very naked. So within the nucleus, DNA is not actually scattered. You understand? So what is the meaning? It means that when you study the cell prokaryotes, correct, right? They say that nucleus is naked and the genetic material lies within the nucleus. Correct? No? Nucleus is naked. It does not have nuclear membrane. But DNA is not randomly just floating in the nucleus. It's not happening there. So how is it done? It is done by some of the proteins. Okay. So these proteins are actually non-histone proteins. Okay. Non-histone proteins. So these non-histone proteins are not as organized as the histone proteins. You will see what is a histone protein, okay, in the next slide. So you just remember that in prokaryotes, the DNA is packed by non-histone proteins. Non-histone means, see, for a histone to be as a unit, it has a specific nature, right? Even though these non-histone proteins are positively charged proteins. Remember this. So that doesn't mean that if it is non-histone, it is negatively charged. No, non-histone proteins are also positively charged proteins, but these proteins are not as organized as your histones. So that is why the nuclear packing is not so well precise when it comes to prokaryotes. Okay, so only some group of amino acids which are positively charged, they come together. So this DNA will start winding around it, and that's how the DNA is packed. Okay, so where is the DNA packed? It is packed in a region called as nucleoid. So in nucleoid region of the nucleus of prokaryotes, that is where you can see that genetic material, that is the DNA. Okay, so usually this DNA is in large loops. It's not in the form of chromosomes. Remember, chromosomes are found in eukaryotes. Okay, just the DNA that is just packed, okay, or just randomly folded. You just remember randomly folded, so that you can see, in prokaryotes. Are you understanding my point? Next one is packing of DNA in eukaryotes. So this is what is in your syllabus. Now you see the difference. Okay, in prokaryotes it's different. It's not so well organized. It is quite random. But the type of protein is non-histone proteins which are positively charged. So this is about prokaryotes. When it comes to eukaryotes, okay. So before I start this, are you guys able to understand what I'm trying to say? Can you give me yes, no? Fast. Please put it in the chat box or give a reaction. That will really help me to understand. You guys are following? Yes, perfect. So some students are saying yes. Others, if you have problem, please feel free to put it in the chat. There is no issue. Okay. Chalo. So now this very, very important topic, packing of DNA in eukaryotes. Okay. Remember that the packing of DNA is done based on the charge that the molecules carry. Net charge, we call it as. What is net charge? Let's say that we have 10 positive and 12 negative. What will be the net charge? Can you tell me what is the net charge of this particular? Okay, in a system. Okay, this is, let's say this is the box. Okay, in this box there is 10 positive molecules and there is 12 negative molecules. Okay, negative charge, let's say, not molecules or charge. Now tell me what is the net charge? If I look in the system, what will be the net charge? Can anybody tell? 2. Exactly. You see, somebody said it's -2. Perfect answer, isn't it? Now, similarly, if I add, okay, a positively charged molecule. Now you know that how the system's charge changes, even though they have negative, but because they have more positive molecules, they gain a net charge of positivity. Now you understand, right? So proteins are also charged. They can have positive charge, they can have amino acids, okay? Amino acids can also have positive charge or negative charge. Okay. Now, when more number of amino acids are in a protein molecule, which are high in positive, sorry, excuse me, they are having more positive charge. So, in the whole protein, it will gain net positive charge. For example, if I have 100 amino acids in a histone protein, okay, out of it, 90 amino acids carry positive charge, and 10 amino acids carry negative charge. Okay, negative charge. That means what will be the whole nature of my protein, histone protein? It will be positively charged, correct, right? So that is what happens. Now, if you look at a histone protein, okay, if you look at a histone protein is again like any other protein, which is a polymer. It is a poly amino acid chain, right? That means you have amino acids that are arranged. Among this arrangement of amino acids, most common type of amino acids that you can find is lysine and arginine. Here it is. Okay, I will not write it. It's very dirty. Okay, you can see lysine and arginine are the two types of amino acids which you can see more abundant in a histone protein. So this, because of the presence of lysine and arginine, so which are very positively charged amino acids. Okay, they are very, very positively charged amino acids. Since their number is very, very high, now your histone protein gains what charge? It gains more positive charge. So that is one thing you have to remember. So histone protein has maximum of which amino acid? Lysine and arginine. Or which amino acids give histone protein a positive charge? You can tell lysine and arginine. You understand? So lysine and arginine you have to remember. So because of them, our histone protein gains more positive charge. Correct? No? So we know that histone is positively charged protein. Now, what about our DNA? Our DNA is negatively charged. Okay, our DNA is negatively charged and our histone is positively charged. Now, what happens is the negative and the positive charge, what do? They attract each other. So they use this simple property of opposite charge attraction to pack the DNA molecule. See how nice it is, right? So in your cell we are talking about. So this just molecular charges just molecular charges are helping the such huge information to be packed in such nice way. Correct? So next we see what is a histone. If I look at a histone, just by the term, it is not like a single bond. Okay. So if I observe deeply, histone is nothing but a protein. And a protein is made up of chains of amino acids. Correct? No? So we have chains of amino acid. Some proteins are made up of a single chain of amino acid. It is called as a polypeptide chain, or a primary protein, secondary protein, tertiary protein, etc. based on how many chains of amino acids are in. Okay. If you see our histone has a lot of monomers. So, for example, which are going from H1, H2A, and H2B. So these are the monomers of, let's call it as subunits. Okay, let's call it as subunits. So small subunits come, different types of subunits come to make one histone. So that means how many are there? There are around 8 molecules which are arranged within a histone molecule. So that since there are 8 ball-like structures like this, okay, which are involved to make one histone, so we call this one ball as histone octamer. Okay, it is called as what? Histone octamer. So let me repeat. What is histone? Histone is a protein. So what is this protein made up of? It is made up of 8 monomers. Okay, 8 molecules. Let's say. Sorry, not molecule. Yeah, 8 molecules. So these 8 molecules assembly to make one functional histone. It is called as a histone octamer. Okay. First, we are seeing what is histone. Okay, and then what is the composition of histone? Histone is made up of 8 molecules. That's why we call this whole unit as histone octamer. Get it? Now, what happens? Let's assume that these histone octamers are very well placed, one beside other like this. Okay. Now, this each histone octamer, since they are positively charged and then DNA is negatively charged, what happens is it starts winding around this positively charged histone monomer, I mean like this octamer, like this. Okay, now you can see here, it starts winding up like this. So this winding happens up to 150 base pairs. You understand this point? Each ball of histone, that is each histone octamer, can wind up to 150 base pairs of DNA. You understand? So now this histone is there. Here 150 base pairs of DNA is wound. Okay, that means like woolen ball like this. It's completely wound. And from there, it has to connect to the another histone. Correct? One histone, then the DNA is stretched, again winding, again it is stretched, like correct, right? Here to here, then again wind, here to here, again wind, here to here, again wind. Like this, it's happening. Now you see, from this histone to this histone, the DNA has to be stretched, correct, right? So this one is also very precisely managed. Now you can see that there is one type of histone monomer called as H1 histone monomer. So this H1 histone monomer is also called as linker. Okay, it is also called as what? Linker histone. So what is the role of linker histone? The linker histone is linking from one histone octamer to the another histone octamer. Okay, so this DNA which links from one histone to another histone is called as what? As you guessed, it is called as linker DNA. So it's very small. Okay, linker. Okay, linker DNA. Got it, guys? Linker histone and linker DNA. So it goes from one histone to another. 150 base pairs, a link. Again 150 base pairs, again a link. Like this, when you see, how do you see? It looks like this, right? Now you can see that there is a thread. There is a thread. Out of this thread, there are histone octamers. And this thread is nothing but the DNA. It looks like the pearl string. So it is also called as beads on the string model. So since in the when you look in the microscope, you can see that it's very nicely arranged beads in the string. Usually you need a very powerful microscope. Okay. So if you look in the microscope, it looks like this, guys. Okay. So this structure, which looks like beads on the string, is called as nucleosome. We call this as what? Nucleosome. Understood? So usually, what is a nucleosome? It's a repeating structure. Histone, wound DNA, histone, wound DNA. Got it? Now, this nucleosome, what happens? What happens to this nucleosome? Further packing happens. So once you have your nucleosome, this is your nucleosome, correct? Now, chromatin proteins. Chromatin proteins. Chromatin protein. So this chromatin protein is a type of protein which is also positively charged. Okay, they are also positively charged. So what they do is they come in between the histone octamers, many histone octamers, they come in between them and then again further condense them. So when they further condense them, it will look like a very thick thread. It will start looking like very thick thread. So DNA looks like fibers. So the fiber-like appearance of the DNA from your beads-like string to fiber appearance. So this fiber is called as a chromatin fiber. No, okay, it is called as a chromatin fiber. So chromatin proteins help in the formation of what, guys? Chromatin fiber. Okay. So this further condensation of chromatin fibers is what gives you chromosomes. So if you see chromosomes, they are colored X-like bodies that are present within our nucleus is called as chromosome. Such an organized structure. Some thousands of genes and millions of base pairs of DNA are very, very precisely organized into respective chromosomes. So even though this topic might seem, okay, positive charge, negative charge, histone, DNA, packing is done. Even though the information is very small, but the role is very, very large, guys. Correct, right? Look at the organization of the information. So precise, 23 pairs of chromosomes or 46 chromosomes in each cell. Correct, right? Each and every cell in your body has the same information. So that is why packing is so significant. Understood, right? So we have come till the formation of chromosomes. Now, if you have any doubts, please feel free. If you don't have and if you're understanding, perfect. Okay. Now, if I look at the chromosome, unfortunately, I don't have the picture. But in the last class, when we were studying about principles of inheritance and variation, I was talking about karyotyping, correct? That means we study the physical nature or the morphological characteristics of chromosomes. That means when I look at the film or a picture, I can see, no, this is how the chromosome appears. So now you might have seen the X-ray films. For example, you might have seen the pictures of X-ray films. Even though if you don't know X-ray, I will try to explain. Okay. You see here, you can see that there is a black dark patches like bands, like a snake. Here you are very dark. Here it is light. Here it is light. Here it is dark. Like this, you can see patches on the structure of chromosome. Now, these patches are not quite random. These dark spots are very high if you look at in chromosome. What are these dark patches? Why are some patches are very dark and some patches are very, very light? So these are called as dark bands and the light bands. Okay. Now, what is the significance of these dark bands and the light bands? So the dark band and the light band shows how the DNA is organized. So in the whole of the chromosome, if you see any chromosome, the DNA packing is not even. It is uneven. At some places, the DNA is very, very, very tightly packed. In some places, it is very loosely packed. Okay. So now you see, based on the packing, we can divide them into euchromatin region and heterochromatin region. What is euchromatin region? Now, whatever the light see, you can see that this light portions is called as euchromatin region. There is a reason why we call this euchromatin. True chromatin. U is true. Chroma, chromosomes or genes. So euchromatin, that means they are useful genes. So these euchromatin genes are what? Useful genes. So how are they useful? That means this is also DNA. This is also DNA. One is useful DNA, other one is useless or functionless DNA, I can tell. So this useful DNA is called as euchromatin. Okay, this is called as useful. Now this DNA is very loosely packed. The reason why it is loosely packed because how does DNA function? It has to become DNA to RNA and RNA has to become what? Protein. Correct, no? So this is the function of a DNA. And this function is happening in which part of chromosome? Euchromatin region. Got it right? Next one is heterochromatin region. What is heterochromatin region? So many arguments are there. So many hypotheses are there. Why heterochromatin? Why? See, if you look at a chromosome, most of the chromosome is made up of heterochromatin. I told you, right? 95% of your DNA is waste, junk DNA, or it is non-functional DNA. What is the even point of having so much waste? Why should we carry so much, information which is not at all used from one cell to another cell? Two things people tell. One is this heterochromatin, even though it is tightly packed and it does not have any function, it gives structural stability for the chromosome. That means the packing of the chromosome is very well maintained because of the presence of heterochromatin. That's what they tell. Okay, so the packing is stable. So chromosome is quite more strong, we can tell. And other people tell that we are not born, we are not the result of today. We have been evolving from millions and millions and millions of years ago. So during this millions of years, many of the genes we have discarded. Like you don't want, for example, your fifth standard textbook. Do you touch it? Or eighth, 10th, do you touch it? It's done. So what you do, you just put it in one side, you stack it. You don't use it, but it's there. So ancient DNA or the DNA which is not useful anymore. So all that is junk. So that is what is heterochromatin. Interesting, right? So that is called as heterochromatin region. So heterochromatin region do not function and they are packed very tightly. Is it clear? Did you guys understand? Come on, guys, say something. You're exceptionally quiet today. Are you understanding, guys? You have to give a reaction or say something yes, no in the chat box. Okay. Can we go for the next topic? That is the search for genetic material. Yeah. Okay. Now I'm getting a little bit confused. If you can hear me. Yeah. So let's go for search for genetic material. This is a wonderful story. I like this topic a lot. Why? Because it's not one time or it is not immediate. Okay. It's not very, very immediate for us to know, okay, DNA is a genetic material. We did not conclude like that. Our conclusions did not begin like that. We had a long story. There's a very, very long story how, DNA was proved to be a genetic material. Now it's a stage wise. It's not, it's like a baby steps that leads to the giant achievement or the giant turn in the history of science. So this search for genetic material is very, very historic in terms of understanding how we do a scientific research. This is very, very useful for students, I would tell. So let's see. Okay. First one is the transforming principle. So during the search for genetic material, the early attempts to understand who is responsible for carrying all this information from one generation to another generation. So this was the first question. So if a parents are there and a child is born, how is the child characters are transferred from parents to the offspring in simple terms? So this was the biggest question. To answer this question, let's go back to our Mendel. So during the process when Mendel's genetics was proposed, he called it as factors. Correct? He called it as factors. And factor was not defined whether it is a DNA, whether it is an RNA or a protein. There is something. We don't know whether it is a biomolecule, whether it is a superstition, whether it is just some pulses or signals, or we don't know. Correct, right? So the beliefs were many and hypotheses were many, but the scientific proof was none. So the first early attempts was the transforming principle. So this is a very excellent, what do you tell, experiment that was conducted by Sir Griffiths. So, sorry, come again. Yeah. Frederick Griffiths, okay, a wonderful scientist, he performed this first experiment. Griffith understood, okay, there is some genetic information that has been transferred, okay, between the cells. So he wanted to find out. So what he does is Griffith's experiment was very, very simple setup. He used a mice, okay, lab mice, and then he used another organism called as pneumonia, okay, pneumonia bacteria, that is Streptococcus pneumonia. So Streptococcus pneumonia is a bacteria which causes a disease called as pneumonia. Okay, so two organisms, one is a mice, other one is a Streptococcus pneumonia. Now, what he does is in Streptococcus pneumonia, we come across two strains. One is a smooth strain, other one is a rough strain. So, what is the meaning of a strain? So, we call anything as a strain when they have a specific or unique characters. Like, for example, if I take a lion and tiger, what do you call? You call it as a species. Correct, no? Because you can differentiate between the characters. In bacteria, what happens? They come in so many different shades, we can't put them into separate species. So instead, we nominate these bacteria into strains, rough strain, smooth strain, okay, etc., etc. So many names are there for different bacteria. Now, why did we give it as a rough strain and the smooth strain? If I look at the cell, okay, the bacteria has a cell, outermost layer, that is a cell wall. Now, to this cell wall, what happens is there is a capsule. So this capsule, okay, is very kind of gelatinous in nature, and it makes the cell looks very soft. If there is this presence of this capsule, okay, so this cells are called as smooth strain. When you look in the microscope, the cells will appear smooth, okay, because of this presence of capsule. Whereas in non-virulent strains, this capsule is very thin or it is absent. You understand? So it is very thin or it is absent. So that makes the cell or the colonies of this bacteria cell look rough. Usually, the smooth looking strains are the ones which cause disease, and the rough looking strains are the ones which are non harmful. Okay, so that is the first thing we have to remember. So you understood that we have smooth strain and we have rough strain, and we understood why it is called as smooth strain and why it is called as rough strain. Out of these two types of bacteria, two strains of bacteria that he has chosen, one is called as virulent, other one is called as non-virulent. Now, what is the meaning of virulent and non-virulent? We shall see. Now, if I call anything as virulent, that means it has the capacity to cause a disease. The meaning of virulency is it can cause a disease in an organism. So this property is called as virulence. So what is this property? Whenever bacteria comes into our blood, it should cause a disease, right? So what is this again, virulency and non-virulency? See, not every bacteria or not every pathogen is capable of causing an infection or causing harm. Why? Because it has to have that certain properties, certain proteins, certain way of living. That is when it is poisoning your body, and that is when the disease is caused. So when that nature is absent, then how can it cause a disease? It's not possible. Correct, right? It just exists or it's eaten by our own cell. Simple and plain. So that is called as characteristics of virulent. Other one is called as non-virulent. Understood? Now, what does they do in this experimentation? He takes mice, okay, and then he subjects this mice with different types of strains. Let's see. First, what he will do, he takes the mice and he takes the S strain, okay. So he takes the smooth strain. First one is what I will, where is it? Yeah, the first one is smooth strain. So he takes the smooth strain and he injects into the mice. So when he injects into the mice, what will happen? Mice will develop pneumonia. What is pneumonia, guys? It mice has lungs, no? So the lungs will be filled by a fluid, okay, a toxic fluid, or so the fluid which is not good for your lungs. So this is called as pneumonia. Gradually they die. So now what will happen, guys? So the pneumonia comes and the mice die. Next one, he takes the rough strain. The second setup, what he takes the rough strain. Now you can see that mice does not develop a disease and it will live. And now what he does is he takes the heat-killed S strain. This is a very important term. Many of us will get confused with. Okay, let's see here. What is heat-killed? Heat-killed. So whenever you hear the term called heat-killed, what you think is, oh, we just boil the whole bacterial colony. That is not happening. That is not is what is happening. What we do is we take the smooth strain, okay? So we take the smooth strain and we make these guys, we subject these guys into shocks. What is shocking? Suddenly, our temperature is around 28 degrees Celsius in the environment we feel, okay? Now, I raise this temperature to 45 degrees Celsius. It's not boiling. I just make it enough hot that they feel very uncomfortable. You understand? I'm not killing the cells. I just raise the temperature to 45 to 47 degrees Celsius. Now, what will happen? These cells will go under a certain shock, okay? And this shock will usually lead to protein degeneration. Okay, protein degeneration. That means the proteins get destroyed at high temperature, or some of the proteins will not function at high temperature. This is what is happening when we heat the bacteria, smooth strain bacteria. So that is why it is called as heat killed. Are the cells living? After heat killing, are the cells living? Yes, they are living. They are living cells, but these cells will not have the capacity to cause a disease. What happened to them? When I heated these cells, they lost this virulence character. Okay, the virulence character, okay, virulent character is lost. okay, is lost during what? During this process. When I heat them, the proteins which are responsible for causing a disease, they get destroyed. When they get destroyed, they cannot cause a disease. You understand? So whenever you hear a called as heat killed smooth strain, you should know that the cells are living, but these cells have lost the capacity to cause a disease. As simple as that. Got it? So they have lost the virulence character now. Now, what do? I inject this heat killed smooth strain into a mice. The proteins are destroyed. Now, what will happen? This mice will live. Simple, no? So, this particular mice will, he gets to live another day. Correct. Next, what I will do? I will take a normal R strain. Okay, let me go for the next slide. I will take a normal R strain. Okay. And then I will mix it with heat killed. Okay. H killed or heat killed smooth strain. When I mix it with heat killed smooth strain and the rough strain, what would happen? You would generally think that this particular bacteria is already non-virulent. This particular bacteria already I heat killed and make it non-virulent. The mouse should live. That's what even Griffith thought. But it did not happen like that. What happened was the mice actually died. So now what happens? Griffith understood something. Okay. Now already the smooth strain cells, they lost the characteristics of virulency when we heat killed it. But when I put with the rough strain, something transferred, some information got transferred between smooth strain to rough strain. So the smooth strain cells taught the rough strain cells how to cause this disease. So the transfer of information happened. So that is what Griffith thought. Okay, something has transferred. And when he told this, everybody agreed, okay, something is moving from this cell to another cell. But Griffith was not able to prove what is the thing that transferred between the smooth cells to rough cells. Correct, right? So that is when Griffith's experiment stops. But Griffith's experiment is a greatest beginning stage for a large outcome. So we will see how Griffith's experiment influenced the further discoveries and further investigation on how the genetic material was discovered. Got it? So I'm sorry if I drag a little bit. I want to make sure that you know the basics very well so that further slides are getting quite complex. So I'll take some time in this lesson because it's a very important lesson. All right, guys, any doubts? We can end the session. Please don't leave. 1 second. Understood? No? Yeah. Instead of asking, I'll ask this poll. Please answer. And thank you very much for attending the session. It's very nice to have good strength. A little while, we'll be back to our studio and we can have a proper session. Okay. A big screen is better, I think. Yeah. So, and as I told you, the people who are feeling difficult, definitely you can reach out to me. I will give individual, I will give individual attention. You know, I will tell you guys where is the problem. I won't mind. I will definitely explain or make a video. So feel free to use this opportunity and approach. All right, guys. Thank you very much. Happy festival and have a wonderful time. See you soon.